Welcome to this session. In agriculture or in farming, what we do is we produce food for people, and we use animals and plants. We need animals and plants to feed us, to produce food for our demands. And the demand for high quality food continues to increase. And so we need animals and plants in the future that are capable of producing high quality food in a sustainable manner. And discussing technologies that allow us to change the genetic makeup of animals and plants to achieve this aim is the discussion point of this session. And it gives me great pleasure to introduce Professor Ben Hayes, who's the director of the ZeroNet Agriculture CRC now, after really we all know him as in his field of quantitative genetics. Professor Ben Hayes has an extensive research experience in genetic improvement of livestock, crop, pasture, and aquaculture species, and with a focus on integration of genomic information and genetic evaluation programs systems. Ben is not only in the research phase, on the forefront of research, but he's also really involved in implementing research in industry, and he has led many large-scale projects which have been successfully implemented in livestock and cropping industries. And so as a contemporary of Ben in the field of animal breeding and genetics, I've had the pleasure of listening to Ben many, many times. And I've always enjoyed listening to Ben because he's capable of actually explaining complex topics in a way that makes it simple and really easy to understand. I'm looking forward to his presentation today, which is about AI for breeding, potential or pointless. Please join me in welcoming Professor Ben Hayes.
Thank you very much, Susanna. And I have to say, Susanna introduced us as contemporaries. That's not exactly right. Susanna was one of the people I really looked up to when I was doing my PhD, so it's a bit of an honour to be introduced by her. So the topic for today is AI for breeding, potential or pointless. And AI means, in this case, artificial intelligence, not artificial insemination. Okay, next slide, thanks. So why do we need new technologies in breeding? In this country, we have an enormous opportunity with our agriculture to meet the demand for protein that is growing around the world. So there was a report done by CSIRO, I think it was, commissioned by AgriFutures, that said by 2030, there would be demand for an additional 65 million tonnes of high-value protein from both plant and animal sources by 2030. That's an enormous demand. So there's that challenge of producing that extra protein. At the same time, though, there's the challenge of climate change. So how do we produce that extra protein with the challenge of extra heat stress, for example? And this little map, this comes out of a tool that CSIRO put together quite a few years ago. It was quite nice. You could put in the city that you lived in, and it would project what the climate would be like based on another city in Australia by 2050. So in Melbourne, by 2050, the climate is going to be similar to somewhere like Warwick. That sounds pretty good if you live in Melbourne, particularly in winter. But if you're a dairy cow, that's a pretty stressful proposition that you have to deal with. So we can't just go on breeding as usual. We need to bring in new technologies. So what new technologies are we going to bring in? I'll actually touch on a couple, but the main one I'm going to focus on is AI. So why AI? It's made incredible advances in some scientific fields. And perhaps the best example is prediction of protein structure. There's a program called AlphaFold, which has just advanced that field many, many times ahead of where it was in the past couple of years. Next slide, thanks. So to go through how we can use AI in breeding programs, I'm going to break the breeding program down into tasks that breeders have to go through. The first one is to set the direction of your breeding program. What do you actually want to breed for? The next thing you have to do is measure large populations of individuals for those traits that you actually want to breed for. Then you can select for them. Historically, we've done that using both the phenotype measures, the measures on the traits, as well as pedigree, who's related to who. These days, we also use DNA information in just about every major agricultural species. Then you have to choose which crosses you're going to make between the individuals you've selected. And once you do that, you get the progeny. From that, you have your new germplasm. In case of livestock, new bulls and heifers. In cattle, new varieties in crops. Okay, so let's go through each of those tasks and look at how AI can help us with those. Next slide, thanks. So the first one is the breeding objective. What are we actually going to breed for? So a little bit of history here. If you go back, say, 15, 20 years, most livestock breeding programs and also crop breeding programs were very focused on breeding for production. Dairy cattle, it was milk yield. Beef, it was kilos of beef. Crop, it was yield of grain. Over time, we've added to those breeding objectives for a number of reasons. One is if you just breed for pure production, other traits like fertility in dairy cattle go backwards, and that's obviously a bad thing. Also to meet welfare concerns, reduce diseases, and also to meet concerns of farmers. So, for example, temperament of cattle became a part of breeding objectives. So if you look at the breeding objective for dairy cattle now, it's actually only 50% production. The other parts are made up of fertility, mastitis resistance. They're putting traits like methane, lowering methane in there, and so on. Next slide, thanks. And increasingly, there's emphasis on sustainability traits like reducing methane, increasing feed efficiency, welfare traits, and also consumer traits. And in Australia, I think we're ahead in some of our industries in doing this. For example, in sheep, in some of the indices, we have meat quality very much in those indices. So those consumer traits are coming in. This is probably the area we haven't applied AI to yet in any form, as far as I know. But there is an opportunity here to use things like large language models to interrogate our consumers, our customers, to get the balance between all these traits correct. To my knowledge, nobody's done that yet, but it's an interesting concept, I think. So next, thanks. Phenotyping, the next stage in the task that we have to do in the breeding programs, measuring our populations for the traits we're going to breed for. This is an area where AI is really making a huge difference. What you want to do when you're phenotyping is measure as many individuals as possible, because that way you capture the most genetic variation. If you're using DNA markers, you want to measure as many individuals as possible so you can get the effects of those markers as accurate as possible and use that in your breeding. To measure as many individuals as possible, that phenotyping has to be cheap. And this is where I think AI can really help us. Next slide, thanks. So some of you will be very familiar with this and probably work in this area. AI can help us by using images that we take of our livestock and crops and processing down those images into the traits that we want to breed for. So that picture in the middle there, that's from one of our colleagues at UQ. That is an AI expert, I guess, works in the AI area. And she's been using her AI algorithms to process pictures from drones of wheat fields where they have different varieties, different genotypes planted out in plots, and she's actually counting the heads in those individual plots to get an idea of how those varieties yield. And that's becoming automated. It's much, much cheaper than having the harvesters go through and actually get the yield mechanically. This can go down to even more resolution. The tractor that you see there, we call it our pheno tractor. It can take images again of these plots, but look at things like disease, susceptibility, or resistance. In the crop world, drones and AI processing of those drone images is becoming more and more routine in the crop breeding space. In livestock, it's coming. Perhaps the best example is in dairy cattle. So in dairy cattle, many dairy cattle throughout the country every day have a milk sample taken. That milk sample goes to a herd recording unit, herd processing unit, and it gets a mid-infrared spectra shone through it. By doing that, they can measure the fat and the protein in the sample, but you also get this spectra that includes an incredible amount of information. And people are using AI to turn that into predictions of how healthy the animal is. That's based on the fatty acid ratio in the milk and so on. Is the animal pregnant or not? So that's another really good use of AI. Okay, next, thanks. So in my scorecard for can AI help us in these different tasks, I'm going to give a big tick to phenotyping. It's actually starting to work really well. The next one down is selection of parents. Yep, next slide, thanks. Okay, so can AI help us here? So just before I go into this, the way we select our parents in most of our livestock and crop breeding programs now is using a combination of genomic, the DNA information, pedigree, and the phenotype information. And in some of our breeding programs, what we do is we select the individuals based on their DNA, a DNA prediction of the genetics they carry for a trait, but that is calibrated on a very large set of phenotype data, what those DNA markers actually do. And we call that genomic selection. Next slide, thanks. Okay, so can we use AI to take in this DNA information and do a better job than our current models of predicting the genetic merit of our individuals, the merit for breeding, the value of the genetics they're going to pass on to their progeny? We're going to try AI just for this case. We're going to try AI in sugarcane breeding. So sugarcane has one of the most complex genomes that we know of. So our genome, we get one copy from mum, one copy from dad. We've got 2 copies of each chromosome that we carry. A sugarcane plant has between 8 and 10 copies of the genome. And it actually varies between families. And just to make it even more complicated, the modern sugarcane plant comes from 2 ancestral species. So you've got 2 ancestors mixed up in there. So we thought this would be a really good test case for AI. AI is supposed to pick up these complexity, interactions potentially between genomes and so on. So let's see how it actually does when it's applied to genomic prediction in sugarcane. I should say as well, we've already established in other work we've done, sugarcane has a lot of non-additive variation. So it's not just when you get an extra allele, you get an extra bit of an effect and that's additive. It's actually quite complex and you have a lot of interactions between genes as well. So it's a good test case for AI. Yep, thanks. The person that actually did this was my student, Chen Song Chen, and he applied a whole range of genomic prediction methods. I'll take you through them in a moment. To this data set we had, where we had 26,000 DNA markers across the genome of the sugarcane plant. We had nearly 5,000 individuals measured for a trait which was disease resistance. Sugarcane gets this disease called smut. It's a rust, which really compromises yield. So you want to give the growers plants that are resistant. Yep. Okay, so we're going to apply a few models here. The first one we're going to apply is our traditional, I'll call it traditional method for genomic prediction, and it's called best linear unbiased prediction. The really key thing you need to know about BLUP is it makes the assumption that there's mutations affecting this trait, in this case smut, but it could be yield or any other trait. There's all these mutations, all these genes carrying mutations smeared across the genome. Lots and lots of mutations with very small effect that add up to give you the differences in the traits. In fact, what it assumes is there's an infinite number of these genes and mutations with an infinitely small effect. That's the mathematical assumption it makes. That's a pretty weird assumption, but let's see how it goes. Next slide, thanks. So Chen Song trialled this against this way of predicting the genetic merit of individuals against a whole bunch of machine learning methods. He tried deep learning methods, convolutional neural networks, random forests. He also tried pretty much the very latest machine learning network method, which is the attention network. So if you've ever used ChatGPT, the attention network is one of the mechanisms that makes ChatGPT work really well. So Chen Song tried that as well. So how did all those methods compare to each other? So what he did was he trained all those models on one chunk of data, and then he predicted in to a validation set, so a set he kept aside, and then looked at how well the DNA predictions lined up with how well the plants actually performed. So let's have a look at that. Next slide, thanks. So this is the answer. Along the x-axis are all these different methods. The BLUP is this traditional method that we use, and then you have the machine learning methods, CNN and attention network. On the y-axis is how well these methods did at predicting genetic merit. This is the accuracy of our predictions. If the accuracy was 0, it means we were predicting nothing at all. If it was 1, we'd be doing a perfect job. So we're somewhere in between. It's actually quite a useful accuracy. But what you'll notice is the BLUP method and the very latest machine learning method, attention network, there's almost no difference there. They're very, very close, which was very disappointing for Chen Song after he spent 3 months on this. So the next question is, these machine learning algorithms work so well for things like ChatGPT, predicting protein structure. Why aren't they working better than our traditional methods for genomic prediction? So I'll just give you a little bit of an insight into that. Next, thanks. Okay, so if you look into the literature, there's actually more than 200 papers where people have tried using machine learning methods to beat our traditional genomic prediction methods, BLUP. And almost all those papers say, they either say, we beat the traditional method, the BLUP method, by 0.05%, or the methods were almost competitive with BLUP. They almost got there, but not quite. So the question is why? Okay, so remember this BLUP method, what it assumes is there's lots of mutations of small effects smeared across the genome, and they all have this little tiny effect, but that adds up to give you your variation you see. Next, thanks. So the question is, really, is this a good model of our trait variation? And by trait variation, I mean for traits, not just disease resistance, but yield, fertility, and so on. So let's zero in on this a little bit, and let's take the trait which has been dissected in most detail, and that is human height. There's studies that have been published now where they've measured 5.4 million humans, us, for height, and they've genotyped them for millions of markers across the genome. So they're zeroing in on these mutations. The other thing to remember is height is very genetically determined. About 80% of the differences in our height is due to genetics. Because we all eat pretty well, there's not much nutrition effect there, it's mostly genetic. So what these researchers find is there's more than 12,000 mutations across the genome affecting height. And the biggest one of those only explains 1% of the variation. So it's not infinitesimal, but it's pretty close. So our BLUP model, although all models are wrong, and certainly this infinitesimal model is not correct, but it's actually not a bad approximation of reality. So that's our thinking on why these machine learning methods aren't giving a great advantage over our BLUP methods for breeding. So in a way it's good, we can keep using our BLUP methods with some confidence. Yep. Okay, so across there, it's a bit of a fail for AI methods applied to genomic selection. Okay, what about for the next part, the crossing part? Next slide, thanks. I'm going to move on from just considering the crossing part, and I'm going to move on to a technology that we've been looking at that in fact does all this selection, crossing, and development of new germplasm together, and look at if AI or a class of algorithm can be useful there. Okay, so to introduce this new method, next slide, thanks. What we've just been through, genomic selection, what you would do is you genotype your animals or your plants, then work out which one has the best genomic breeding value, and then you line up those individuals and select the best for breeding. So in this case, you'd select the bull with the highest protein, EBV, to use to produce progeny. But once you've got all this genomic data, which you have to remember comes from having a very large phenotype data set where you calibrate your DNA markers, that's the origins of these genomic breeding values. Once you've got that data, you can use it in a different way. Next slide, thanks. Yep, next. You can actually peer into the genomes of those bulls, and you can look to see which chunks of genome they carry. And you can start to ask the question, across the population, what's the best chunks of genome there? So the bull with the best breeding value overall, he carries a lot of good chunks across his genome, a lot of mutations with good effect, but there's a few cases, and I've highlighted in red here, or one case there, where he carries a bad chunk of genome. Whereas another bull, who's not so good on his overall breeding value, he has quite a lot of bad chunks of genome, but he's got just one or two good chunks. What we'd actually like to do is put together all those good chunks into the one individual. And you can identify these chunks of genome once you have these DNA-wide markers. So next slide, thanks. Okay, so you can think of now a population. You zero in on the genomes in your population and you pick out all the good chunks of genome. Yep, next, thanks. And what you want to do is bring all those good chunks of genome into one individual that's going to be your ultimate individual. And it should have higher yield, better performance than other individuals in the population. Next, thanks. We've actually looked at this, and other authors have looked at this in various crops and dairy cattle. The predictions are when you stack up all the good chunks of genome, good haplotypes in dairy cattle, you get a performance that's 6 times higher than the best individual in the population today. That might sound like science fiction, that you can actually do that, but if you look at dairy cattle where we've come from in the 1940s to today, we have actually already achieved that kind of increase. So we could go further than that. And this has been done in wheat, in chickpeas as well. And the fold increase in performance you can get to is quite substantial. Next, thanks. Okay, the problem is you have to design a breeding program to actually get there. And that's where we use this genetic algorithm. Some people class this as a type of AI, some don't. Doesn't really matter. This is the algorithm we're using to choose those parents out of your total population that will stack together all those good haplotypes. The genetic algorithm's a good one to use because it can efficiently sort through many, many combinations. And for example, I think the example there is if you want to choose 25 crosses out of 100 possible parents, you've got billions and billions of possibilities. So something like a genetic algorithm or some other form of AI is necessary here. Okay? We've actually been doing this together with a wheat breeding company called Longreach. So we took all their germplasm, we looked for the best haplotypes they had, best chunks of genome, and started to make the crosses, to put them together to create these ultimate genotypes. We had a bit of help here. We used something called speed breeding, which is where you grow up these plants in a glasshouse with the lights running 24 hours. And that way you can cycle through, say, 7 cycles of wheat breeding in a year, whereas it usually take you a year to do that. So it can really speed up that process. The good news is, yep, next slide, thanks. Yep, next. Those varieties, those new varieties, stacked varieties, have actually been planted out in the field and gone through a field trial. You can see the results here for 4 locations around Australia. And the new varieties are the blue, the old Czech varieties are the red, and the y-axis is the yield of those varieties. So you can see these new stacked up varieties are outperforming the old Czechs. So at least for this field trial, this idea of stacking using AI, or at least our genetic algorithm, has worked out really well. And we're now actually applying this to all the crop species we work with. Yep. In livestock, it's a bit more tricky because the whole thing works on having large numbers of progeny to get the recombinations that actually stack up the good chunks. In livestock, where you just get 1 offspring per cow per year, that's a bit tricky. But you can start to use technologies like IVF, in vitro fertilisation, and even in vitro meiosis is starting to be thought about, where you have a sperm and an egg in a petri dish, produce embryo, and then differentiate the stem cells in that embryo again into sperm and eggs, cross them again, and go through your breeding cycles really rapidly like that. Some companies are working on that at the moment. Next, thanks. Okay, and aquaculture, this should work really well because in aquaculture you can breed millions of offspring per mating. Yep. Okay, so for this combination of selection, crossing, and developing new germplasm, I hope I've just convinced you, shown you a way of using the technology that can really speed up our current breeding programs and produce new varieties, new germplasm that is going to increase genetic gain towards our breeding objective. Yep. So just to conclude, AI is showing great promise for some parts of our breeding program. Not all. It doesn't seem to help for genomic predictions, but for other parts of the breeding program, phenotyping, new breeding program strategies, it really seems to help. I do want to say though, just go back slide, thanks. This is all underpinned by data. Unless you have data to drive the traditional approach, the new AI approaches, you can't get anywhere. You really need very large data sets, records of your animals or your crops for the traits that you're breeding for. That is the whole key to it. And if you think about the AI methods, if you read a little bit about them, something like ChatGPT has 1 million parameters that it's trying to estimate from a data set. It can do that because it uses the entire internet for training, which is billions of data points. But if we try and train a method that has millions of parameters on a data set of 30,000 animals, that's not a may not be a particularly sensible thing to do. How do we get these enormous data sets? Particularly for traits that are becoming important, like sustainability, like consumer acceptance, consumer traits. How do we actually build these big data sets? And what I'm suggesting, and as many others have suggested and have actually done, is that we should collaborate internationally to build these big data sets that we can all then use. And I'll just finish by giving you an example of this that has worked really well. Yep. So this was a project that was actually led by Pete McGilchrist here at UNE and driven by Rod Polkinghorne on consumer quality, eating quality of beef. This was a pretty amazing experiment. There were over 3,000 beef samples eaten by a consumer panel and scored on things like tenderness, juiciness, overall liking and so on. At the same time, there are experiments going on in the USA and in Ireland where they were doing the same thing. The aim of the project was to come up with genomic breeding values for these traits so that you could genotype a bull calf and predict how well the steaks from his progeny were going to eat, how good they were going to be for consumers. So we had about 3,000 of those records here in Australia. We knew that would give us moderate accuracies, but it was actually Rod said, why don't we team up with Ireland and the USA, put all our data together to come up with these genomic breeding values for consumer eating quality. And this actually worked really, really well. You can see the graph there, the different traits. On the y-axis is the accuracy of those genomic breeding values, how well the DNA markers were predicting the true breeding value of the animals. The blue is the Australian results, and you can see for most of the traits, you're getting a substantial jump up by combining the data. So that's worked really well. Yep, next slide. Okay, just to finish up on one point, the flip side of this is that really high quality data that farmers collect, breeding programs collect and so on, this data is going to become incredibly valuable. If you look at what the AI companies are doing, they're now looking around the world for high quality data sets that they can take, process through their algorithms and add value to, come up with some new app or something that we can use to make decisions. So really good data just becomes even more valuable than it was in the past. So I guess I'd like to leave you with that point, that new AI algorithms are wonderful, but underpinning it all is this high quality data, and we should work together to put these data sets together. Thank you. Just finally, thank you to everybody that worked on what you saw here. Sorry, you can go back, you can cut it down. Yep.
Go back to the, just give you this. So,
Okay.
do you want wood?
Okay, I'm fine, thanks.
Well, again, join me in thanking Ben for his outstanding presentation, please. Before I introduce the panel members, I'd like to actually give people the chance to ask questions to Ben's presentation now rather than at the end of the session while it's still so fresh. And I have one question here on the Slido by Lloyd Thompson. Does the research on high levels of omega-3 and speckled park cattle change our focus on breeding for eating quality? A very specific question.
It is a very specific question. So omega-3 is obviously very valuable and is worth breeding for. Interestingly, there's a company in New Zealand. They do something really nice. They breed the lambs to have higher... Is that gone off?
That's just gone
off. It's back on now. Yep, good. They breed the lambs to have high omega-3, but then they graze them on pastures that have high levels of omega-3. And they put that together as a package to produce high omega-3 lamb. That's a really nice concept. So yes, you can breed for omega-3, but again, you've got to measure it in thousands of animals, and then you can certainly breed for it. Yep.
And I have a question by Elena Denavi. I was wondering how we can breed livestock for top trait performance while avoiding inbreeding and line breeding.
Okay, so yes, and what's happened in the dairy industry, sadly, actually, is as genomic selection has been applied, it's been possible to identify really, really outstanding bulls at a very young age, and they've been used very aggressively in the industry to breed sons that then produce semen for dairy farmers around the world. So inbreeding has actually gone up. This is a shame because you can use the genomic information. When you have the DNA information, you can see extremely accurately how related the animals are to each other. And there's good ways of using that information to manage your inbreeding and the rate at which your inbreeding goes up. We just have to actually do this in our breeding programs. And some of the breeding companies are now doing this and some aren't. And maintaining that diversity is particularly important when you think about future traits coming online. We need diversity for things like methane emissions levels, feed conversion efficiency, and so on.
And now I have a really interesting one, because many of us are scientists here, and Mina is asking, what do you think about future of AI on a scientist's life? Is AI a threat or assistance for scientists?
Oh, that's a tricky one.
That's a tricky one.
That's tangled. How am I going to answer that? Well, I will admit to help using AI to help me write some reports. And it's very, very good if you put down the key dot points and you feed it that and you say, write a report for me. I will admit that. The thing is, you have to check it extremely thoroughly afterwards to check it hasn't, they call it hallucinated, come up with something strange. You do have to be extremely cautious using it. One thing to realise is everything you type into these chatbots and so on gets uploaded into the internet and then it's publicly available. So if you've got a scientific discovery you've just made, it might be the last thing you want to do. Yeah. So I guess exercise caution and understand exactly what the AI is doing for you.
Great.
Do you want to answer that as well? Or?
No, my point was like for automated processes, just to make life easier for us, just a repetitive type of tasks. I think we should just learn it as a tool. But if it comes to creative thinking and innovation, then it's still people's mind, in my view.
Yep, absolutely.
I don't know so we have some microphones. If you maybe just take a seat on the last seat there, Ben. But firstly, again, join me in thanking Ben for answering the questions here. I might pop other questions to you during the panel discussion, and what I do now is I'll introduce each panel member, and if once I've introduced you, that you just sort of take your seats along the row, and then I have questions in regard to breeding objectives, phenotyping, and use of genotypes in commercial environments. So there's 3 different areas, and I encourage you to put questions into Slido, and then we have, discuss these questions or address these questions in between rather than all at the end. So feel free to put questions in, and I will look at them and see when they seem appropriate or when we have time. So I'd like to start. Michael Crowley is our first panel member. He's the General Manager of Meat and Livestock Australia, or MLA. Michael has held a range of commercial positions in the meat and livestock industry, including being the CEO of Herefords Australia. Michael joined MLA, where he's now the General Manager, in 2009, when he managed the Meat Standards Australia program. After a stint in Brussels, he came back to MLA in Australia for various positions, and then last year, Michael took on the role of General Manager of MLA. Please join me in welcoming Michael. Our next panel member is Professor Nina Mitter. She's now the Deputy Vice Chancellor Associate at Global Research at Charles Sturt University. Nina is globally renowned for her leadership of innovative platforms, namely RNA-based biopesticides for sustainable crop production. And I'm looking forward to learning more about these things here in our discussion today. So she's also worked on clonal propagation of avocado using plant stem cells and nanovaccines for animal health. With the increased scrutiny on the use of chemicals as crop and animal disease control agents, Professor Mitter is focused on developing clean technologies for the agriculture of tomorrow, focusing on animal plants health. Thank you. Our next panel member is Dr. Sonia Dominic. She's a principal research scientist at CSIRO. With CSIRO Agriculture and Food, working on sustainability credential and indicators in the agriculture industries. She focuses on decreasing the complexity of processes in agriculture with the aim of reducing the cost associated with compliance and enable trusted data streams that ensure global market access. Sonia is also a quantitative geneticist, and I've had the pleasure of collaborating with her on breeding-focused books and workshops. And Sonia held various leadership roles within CSIRO and has applied experience in the aquaculture and the livestock industries. Please welcome Sonia. Our remaining panel member is making down the stairs. It's Dr. Steve Jeffries. He's currently the principal of Jeffries Ag Solutions. However, Steve is a highly accomplished plant breeder. Steve is the principal of Steve Ag Solutions, which provides consultancy service to agribusinesses. Prior to this, Steve was managing director of GIDC and the founding CEO of Australian Grain Technologies, Australia's largest and market-leading plant breeding company. Steve is also currently the chair of Rice Breeding Australia. Please welcome Steve. So as I mentioned, so thank you very much. Do we have enough? Only one fine? Where do we sit? Just one. The other one died. So sharing is the game here. In regard to breeding objectives, I'd like to start with Michael and asking a question for him. Can you basically give a brief background of the meat industry, the red meat industry, in regard to their challenges and opportunities, and where do you see the shift in the components of a breeding objective in the red meat industry emerging in the future?
Yeah, thanks very much, and good afternoon, everyone. Look, we operate in a global marketplace in the red meat industry and supply over 100 markets around the world. And one of the biggest challenges I think we have as an export-focused nation, even though our domestic market is our largest and most valuable market, we operate in a global context, even domestically. The changing expectations of our customers around the world, as well as consumers, is going to shape the way that our production system needs to respond. Ben spoke about the work in Ireland and the US and Australia, and that gives us a lot of prediction capability for how we select for eating quality outcomes for consumers. So at point of sale, making sure that our products meet and exceed a consumer expectation is absolutely fundamental. We're also seeing additional requirements being put on us from either importing country requirements from different market segments, so our supermarkets domestically, supermarket chains internationally, multinational food service companies, looking at different criteria around animal welfare, environmental sustainability, and nutrition, health and nutrition. So when I think about how does our production sector need to respond, we will see those specifications impacting pricing signals, and we need the tools for our livestock producers to respond, and genomics is a very important component. And I think we have a huge opportunity to connect with what our customers and consumers want and design breeding programs and breeding livestock with a market destination in mind. And the genomic tools and technologies available to us are a big enabler for that.
Great, thank you. Sonia, you're sort of here looking at the aquaculture industries, and could you give us an overview of the challenges and opportunities for genetics and aquaculture and their impact on breeding objectives?
Yeah, thanks very much, Susanna, for inviting me to give the aquaculture perspective. And I assume that the majority of people are less familiar with aquaculture production. So in general, as Susanna and Ben have set the scene, that protein consumption increases, and aquaculture is certainly a big opportunity to meet that, in particular with the decreasing fish stock. So taking aquaculture species into more controlled environments is a big opportunity to increase sustainable protein production. In Australia, we don't export a lot of seafood. We actually import more seafood than we farm. So again, there's actually a big opportunity in that space. In terms of species, it's a bit of licorice all sorts. It's something for everyone. There's from crustaceans, other mollusks, other fish, fin fish, and also algae. And within that, you got the filter feeders like oysters that don't actually need any input, to high input species like Atlantic salmon. And also in terms of breeding programs, they vary greatly and range from production where they actually use wild-caught broodstock over sophisticated breeding programs that use genomic selection or marker-assisted selection, all the way through to gene editing, which has now been achieved in Brazil. In terms of traits, aquaculture species focus very strongly on growth, but with the changing climate, certainly the emergence of disease challenges becomes more prominent. So certainly disease resistance and therefore climate resilience is certainly gaining in prominence.
Yeah, thank you very much. It's in a way, it's quite different in aquaculture. It's such a diverse range of species still, and there are different stages of development for producing food for agriculture, whereas in our livestock industries and plant industries, we're probably in horticulture, we haven't got that variation maybe that we have in aquaculture at one point. Yeah. So Jeffrey, you've been very successful in developing wheat varieties yourself, and so you're very much aware of the important traits, and in the past historically it's been yield, and then maybe disease traits. And so where do you see will be the main selection emphasis to be placed in the future in plant breeding and crop breeding in particular?
It's still yield.
It's still, yes.
It's still yield. Speaking as I wear my previous hat as a CEO of a commercial plant breeding company, yield still is right at the very top of all the list, and there's decisions by a grower to choose which variety to grow. However, if I think more broadly of the grains industry at the moment, and you look at where my previous full-time job at GRDC and where we were putting the greatest effort in breeding, and that was into pulses. And the reason for that is driven by a few things. Firstly, input costs. So cost of fertiliser is becoming probably, particularly nitrogen, is becoming by far the greatest and highest risk cost faced by farmers. grain farmers. The other factors are sustainability related in regard to, again, nitrogen fertiliser is the highest emission generating part of the whole grain production cycle. And the third part of it is here on the East Coast, it's the grains industry, the domestic market absorbs most of the grain. But if you look at Western Australia and South Australia, the bulk of that is exported. And we're competing, particularly in wheat, we're competing with Black Sea, Russia, Argentina at a lower cost. And it's really important that we are pitching at a higher price end market. So again, protein is a fundamental part of that, and I'll talk about the other parts later on. But protein's a fundamental part of that, and a farming system built around a legume rotation, and we heard it this morning from Sue, built around a legume rotation is really critical. And like the Western Australian farming system at the moment is based around a wheat canola rotation, which just takes nitrogen out of the system. So that's it. When it comes to traits more broadly, look, disease resistance is absolutely critical, and the reason for that is, again, cost of chemistry. It's about deregulation or regulation of chemistry, and it's also about consumer behaviour and consumer attitude to things like residues in grain. So plant-based resistance or alternative ways of doing things that don't have residues, and I'm sure Nina will talk about that, is really important. And probably I touched on quality, and I'll skip over that one a bit more. And the last one is really related to climate change. And so, there's a much greater focus across the breeding effort and the pre-breeding effort in the grains industry on things like heat and cold, more so than they've been done before. I see Ben nodding his head. So.
Yeah, thank you very much. You've actually alluded to my next question for Nina. And Nina, in preparation for this session, gave me another term. It's maximum residue limits. It's nil MRLs now. So, Nina, how do we address the issue of chemical withdrawals, residue resistance, and pesticides emissions as the world is setting net zero targets for almost nil maximum residue limits?
Thank you, Suzanne. And before I answer that question, if I can indulge in listening to all the things that have been talked about, I just wanted to say, listening to all of these, I'm a strong proponent of borderless innovation. And that's not just about me crossing borders here from India to Australia, but about science and platforms. The things that we have talked about today, whether it is aquaculture or grains or meat and livestock or horticulture, I think the platform technologies that we are developing cross those boundaries, and Ben gave some very nice examples, and that's something that we can carry forward, whether it is genomics, genetics, across the industry, across the ag industry, and that was really nice to hear as well. Coming to that specific question, and this stems, I think some of it was answered by Scott's talk in the morning, and Sue also mentioned about, less pesticides, but at the same time, we have the data that we do need them. We do need crop protection measures. There are 8,000 products at the moment, chemicals registered in Australia. We know that Deloitte report that Scott talked about, that there is a 31.6 billion these crop protection products enable that value, and that's a 53% increase in the last 5 years. The question now we are having is, how aggressively do we need to look for alternatives to chemical pesticides, not as replacements, but to reduce the usage? And that's really important thing to keep in mind, because crop protection is important. Judicious use of pesticides or responsible agriculture, as I call it, is very important as well. So when we talk about some of those measures, and you talked about maximum residue limits, what's happening across the globe, there is now a big momentum on almost zero maximum residue limits. That is, when you are exporting a crop or a food produce or something outside, international, and 70% of our food produce is exports, the countries are putting very tough restrictions on what chemical residue it can carry. And that's why there is now very much a need for exploring those biosolutions to come in various forms, Suzanne. Otherwise, we will be in a spot where we cannot. And we also know, in addition to these maximum residue limits, and Scott again touched on that, there is a chemical withdrawals. The issue of not only residue, but resistance development or toxicity, like we had 12, I think, banned last year by APVMA, a group of category of chemicals which Scott mentioned. So there is that side as well, which again reinforces that we need to look for alternatives, and those alternatives, that is where maybe some of the researchers and the innovation comes in, not only in terms of new chemistries, but also in terms of biopesticides. We'll be happy to talk more later.
Yeah, we'll come to that question later on. So I think the next question is probably for Ben as a starting point. It follows, if you pass the microphone over, it's by Michael Aldridge, and I'd like to just raise it now. Should data, because it follows on the point of what Nina said, of like being open and sharing and collaboration of data among researchers. And Michael Aldridge asked, should data owners consider selling or leasing data sets to AI companies? Is this a route for breeders to be compensated or incentivized for phenotyping?
Okay, I think actually the key principle is, if the data is collected in farmers' fields and so on, ultimately the farmers own the data. So it goes up through the research organisations and so on. And then it gets to the AI companies. If the farmers still have some control over the data, they can kind of push those companies to make sure that that data is used to have some benefit for them, I think is the really key point. I don't think we're going to get very far as an industry if, for example, farmers' data is just hoovered up and given to the AI companies. We have to close the loop a lot better than that.
Yeah, I think what we talked about this morning, regulatory science, and actually just we farmers are not equipped in regard to probably the legal and policy and all sorts of other business decisions type of thing, in my view, to really come up with a framework there. So I think we need more support from other groups to help to tackle this one, because I can see the benefits that would be huge. It's just the framework around it has to be set up to really cover all loopholes.
Yep. Yeah, there's two extremes, of course. You can just give your data away. The other is you can sit on it, never goes anywhere, and then you don't build these amazing apps and so on that are useful. So you have to come to somewhere in between.
A win-win middle ground in a way.
Yes, that's right.
Yeah.
That's right. And I would have to say, in my experience, the collaborators, the herds we've worked with and so on, they're incredibly generous in giving their data as long as it benefits the industry. So as long as we can make that clear time and time again, I think it's okay.
Yeah. I have another question for you, Ben, on phenotyping. Because you mentioned speed breeding, so it's a buzzword in plant breeding, I've heard it. So are there any side effects when putting wheat cultivars through the speed breeding process?
That's a good question, and there is actually a few papers where they've looked at that, particularly if you start selecting the plants during the speed breeding process for traits that are correlated with yield. Do you end up at the end of the process with the plants you actually want? I think a better approach is actually to measure the plots, do very well-designed field trials and so on, record that data, genotype those individuals, and then when you're speed breeding, select on those DNA markers, on those genomic breeding values. Okay. So you don't end up going off in some direction that results in a plant that's not well fit in the field.
Yeah, I mean the speed breeding.
Do you want to add
Jeffrey, would you like to add, please?
Ben's exactly right. When you do what they call speed breeding or accelerated single seed descent, which has been going on at some levels for about maybe 70 years, you're forcing the plant to go through a full development cycle in a tiny window. So you end up with plants that normally grow this high. They're about this high and they produce 1 or 2 seeds. The phenotype that you're seeing during that process is nothing like what you see in the paddock and what you see in... And so it's almost impossible to do any real phenotyping. The power of genomic selection, however, is... Through that speed breeding process, you're dealing with individual plants. So with individual plants, you can take DNA out of those plants while they're growing, and you can do selection based on the prediction of their performance, and therefore you can cycle around really fast. So that's where the power of the whole process really, really starts delivering.
Yes, okay. So, Jeffrey, given that you've got the microphone, I might as well continue asking you a question. Salah this morning demonstrated new technologies to automate phenotyping in crops, and Ben also has included them in his presentation. I'd just like to have your opinion on the use of these new technologies. Are they ready to be applied on wide scale, large scale operations in the field every day, consistently, so that we can use them for our breeding programs?
Well, some have been applied for 50 years,
right.
While others, like some of the ones that Ben talked about, are, from a crop breeding point of view, are either just getting applied or are very soon. So phenotyping is a huge challenge in a commercial plant breeding program. Take the company that I came from, AGT. It produces or manages over 300,000 field plots for yield every single year. Over 300,000. It's massive, massive. That's across 50 different locations. So the phenotyping challenge of harvesting that, you buy a harvester, for example, that's $500,000 to $600,000 for a plot harvester, and it can harvest between 600 and 800 plots a day. And if you're doing 300,000 and you want to do that all in a say, just in the month of December, this is a massive, massive thing. So any help, that's just yield. But the breeders need to look at it for plant type and maturity and lodging and disease resistance and God knows everything. Every little piece of help that you can do there means you can do more with less, and that enhances your rates of genetic gain. So using image analysis, either whether it's a drone or it's a phenomobile or a satellite or whatever, and you can take some of that workload away, you can do it more faster, you can do it more rigorously, absolutely bring it on. And then in the lab, there's, when I talk about technology that's been used for 50, 100 years, NIR for protein is AI, isn't it? It is, because it's not actually measuring protein, it's reflecting light off the bond angles in the grain, or in the bond angles in the compounds in the grain, and building against a model with huge data set that Ben talks about of actual protein measurement, and gets a prediction of what protein is with about a 97% accuracy. So that sort of thing is now going into doing image analysis, for example, measuring grain, taking a picture essentially of whole grain. You can then get an algorithm predict screenings percentage, test weight, grain size, black point, and then you can actually predict functionality. So commonly in barley breeding programs now, they're predicting mould extract, which normally would take you have to germinate the grain, then you have to extract the malt out of it, you have to measure that. That's a huge expensive process. They can get a prediction of this by simply reflection of light against a whole grain. Then they get to use the grain in the breeding program. So it's massively powerful. So the range of options of using AI, machine learning, and basically algorithm-based selection models is enormous in breeding. I think it's almost endless.
Great, thank you. I'd like to follow up with Michael on this question of automating phenotyping, basically, because we do need phenotypes for our breeding program. So, Michael, you're looking at the red meat industry in particular up north, very extensive production systems, and then you need information also from the boning room on carcass quality and carcass value. Yeah. So there's different areas where I can see automation of getting information that's useful. So take your pick. You can't answer everything, but just whatever you will see as most important in that regard to see automation.
And I think when we look at where our highest quality data comes from, it's been through co-investing with either breeders, breed societies, and state governments, where we've been running fully pedigreed, full performance recorded animals and following that, the progeny of those animals all the way through to collecting the carcass data, and also building in lab measurements on top of that. So our very high quality data is also very expensive, and it connects with the question that Michael asked. Because when we get into extensive production systems, some of those traits are hard to measure because of the scale of some of those operations. There's a cost attached to collecting high quality information, but as a result, that does underpin our ability to have improved accuracy in the genomic prediction. Those projects are very expensive to run as well. So what we need to do is have the right incentives in place for ongoing collection of phenotypes. And there's a good example of where there is a disconnect that also creates a big value opportunity. We graded over 3.5 million beef carcasses through the MSA grading program last year, and there's less than 1,000 carcass records going back into breed plan. So we have a disconnect between making a genetic selection at a seed stock level and what the outcome of that genetic selection was for the commercial producer over the hooks. And there's hundreds of dollars difference in value in an over the hooks measurement just using quality and yield. So there's a huge upside potential for getting that information to flow. When we look at small stock and in the sheep industry, we've funded for years reference population. We've got the MLA resource flock. We had the predecessor of that was co-funded through the CRC, and it developed huge amounts of data that enabled the use of genomics, and continues to be high quality input data for a lot of the key production traits. But it's the hard to measure traits that are the most expensive and yet are connected to where we can create the most value. So eating quality again in sheep meat is a big upside opportunity. We now have mandatory EID for lamb and for sheep and for goats, and it's going to create a whole lot of new data capability to connect the commercial outcome and those predictions all the way back to how we're making genetic selection and how we optimise the production of those animals through the supply chain, because we're seeing increasing cost of production and going head to head with improving quality of other major beef producers around the world, and how do we differentiate ourselves. So some of those hard to measure traits are also going to require a significant amount of investment, a new technology where we've got green feeders measuring methane in both the feedlot sector and extensive grazing. We've got that working now across beef and sheep, and looking at what are the commercial AgTech solutions to collecting those traits that are more cost effective than doing it at a lab or at a research level, and commercialising that technology. And there's value in producers investing in that tech is an opportunity for us going forward. And I think that comes back in the form of the right price incentives from supply chains who can then use that information to differentiate their brands. And brand equity will be the significant driver of the addition in value for livestock production. And we can also credential those livestock and feed that information forward through the supply chain with the data capability that we now have. So I think there's some pretty big upside opportunity, and there's a few significant challenges in there too.
Yeah, great. And thank you. And if you pass the microphone over to Ben, because I've got a very generic question here. But from Elena Denavi, and how much more gain can we expect from current and future tools? And are there areas where further gains may be limited?
Okay, so genomic selection has doubled the rate in genetic gain in dairy cattle. There's no doubt about that. So I think we can go close to that in some other industries as well. I think the key, we can certainly increase gains, but I think a key point is that gains are going to look different in the future. We're probably going to have perhaps the same rate of gain for production, or maybe even a little bit less, but we're going to have more emphasis on the consumer traits, end-use quality traits, heat tolerance, and so on. So for the overall breeding objective, these technologies like we've been talking about will accelerate gain, but for the individual traits, there'll be more of a balance that you'll see in the future.
Yeah, and I might add an example that comes to me from the pig industry in regard to selection for litter size. And a huge gain has been made in litter size, and there is more genetic variation that we could use. However, certain groups say, no, is there a limit? Because the sows that have larger litters require much more care of the large litters. It's much more difficult to manage them. So in Denmark, it may be possible to manage these type of litters, but in other countries where staff are not as trained or so, or where staff costs are too high, it may not be possible to manage these high. And so there is discussion about should we limit the productivity to certain levels just for these social and welfare concerns.
Yeah, I think just elaborating on that, and elaborate on what Scott said before, I think we have to get, this is a challenge to our breeding community, we have to get better at measuring fitness of our animals and plants. Because as we go faster and faster with our breeding technologies, genomic selection, speed breeding, in vitro meiosis and so on, it's easy to go very fast for production, for example, but unless we measure the fitness of our animals and our plants, we could well see a deterioration in that fitness, and things like you were mentioning, Susanna, unable to rear the full litter.
Yeah.
So I think in the future we have to get a bit more focused on fitness. Exactly how you define fitness and measure it with a drone or some new way of phenotyping it is a challenge for our industries.
Yeah. And I have now 2 questions for aquaculture. So given that we have so much time left, Sonia, I'll pass the phone over to you. So the first one is heat stress accelerate the reproductive stage in some species. What are some of the strategies to deal with expected climate change in aquaculture?
So it depends, I guess, what sort of aquaculture system you're talking about. So there's like salmon and tuna, which are sea-based cages, where they have their commercial production. Then there can be pond production of various aquaculture species, and there can be also reticulated aquaculture systems or RAS systems that are fully controlled and enclosed. Certainly, climate change can have an effect on all of them. We've just recently seen devastating effects for the salmon industry through disease outbreak, which is clearly caused by the warming waters. But also, if you say for pond-based aquaculture systems, say in northern Australia, with in the dry season and evaporation, the evaporation rates are huge, and the amount of water that is actually required to either top it up or not produce during the dry period is quite substantial. And yeah, then there's also the RAS systems, and I actually encourage you up there. We got a gentleman that I talked to during the break, Matthew Goddard from Aquaculture, who's actually looking at RAS systems and is trying to make them more holistic, because it's not just about putting animals into a controlled environment, but really working with a very complex production system and making it something that supports production. So yeah, there's a huge variety, but certainly all of them will have to consider how to manage climate change in a sustainable way.
Mm-hmm. Thank you. And a follow-up question for you, Sonia, because it comes from Gunilla, and aquaculture has been facing significant challenges regarding social license, what you've just touched on. And how can selective breeding science help in this area? How can selective breeding help in this area of addressing some of these issues that the aquaculture industries are facing?
Yeah, I think breeding is certainly part of the puzzle to actually ensure that sustainable production needs to consider all aspects and all levers of production to actually ensure that it's ethical and sustainable. Breeding is an important part of it, so certainly animal health is a big consideration. Also, the environmental impacts in terms of the inputs through feed, potential environmental impacts through the production system, and what was the other thing? Welfare in terms of fish are also starting to be considered as a sentient species, which previously they haven't. And yeah, there is clear indication that certainly stress impacts their production levels, and breeding can certainly play a role in that. There's distinct differences in behaviour and stress resilience in different fish, for example. So yeah, certainly wherever there's diversity, breeding can be one of the aspects that can help.
Yeah, thank you. I'd like to follow up with Nina on health in regard to using genetic tools to improve health of animals and plants. We can firstly focus on the genotype of the host, but we also can focus on the genotype of the pathogen. Or we can use really other technologies that Nina is the expert in to manage health of animals, or of plants in this case, better. So could you just elaborate on was it RNAs? Yeah, RNAs-based biopesticides for crop and animal health.
Thank you. Thanks a lot. And as we've been, hearing from this panel, it's this combination of tools which applies to horticultural crops as well, or grains, or aquaculture and livestock. When we talk about, especially in the horticultural sector, if I was to say, once again, breeding, trait improvement, the traits can be different because it's the colorful world of fruits and vegetables. So we look at consumer traits, which become very important, and the tools such as gene editing has been mentioned. Once again, these technologies have come a long way since they started, like when CRISPR started with gene editing, to what it is now, how it can be more efficiently delivered, how it can be delivered without using agrobacterium. So all those things are step changes that happen and brings phenotyping into the mix as well. To your specific and my passion on RNA-based biopesticides, so this is based on RNA interference or gene silencing platform. RNA interference was discovered 2-3 decades ago and became the most talked about thing, and it's a conserved mechanism in all eukaryotes. And I started using RNAi to develop transgenic or genetically modified crops for disease resistance, so having that, and it works. The question was adoption, regulation, community acceptance, social license. Not only that, you cannot transform all crops and get those transgenic traits. I tried with avocado and failed miserably. So the question then was, how do we make it, in my mind was, what's the next step in innovation? And this is about 10 years back when we started looking at, can we spray RNA just as a fungicide or a pesticide on crops and still get that protection? And that has really evolved further. These are RNA-based biopesticides. So if you, thanks or no thanks to COVID, people now talk about RNA a little more, the RNA vaccines for human health. This is about using a double-stranded RNA as a spray on crops, specific protection. So you design, as Suzanne said, you look at the genomics of your pest or pathogen. So you look at its genetic makeup to design your dsRNA. And it's almost like nature versus nature, because you design the dsRNA from the pest or pathogen to kill the pest or pathogen. You can just spray it on your crops, and it is crop agnostic, but pest and pathogen specific. So it works on grain crops or on horticultural crops. We sort of dipped our toe in animal health as well, along with Ben's team. But the key point, once again, here is, we can make it specific. So it's specific, so we don't harm our bees and butterflies. It's safe, but it is not a silver bullet. It's just one of the tools, as I mentioned, whether we look at gene editing or RNA-based biopesticides or new improved chemistries or breeding for disease resistance. All these things have to work together. The advantage for me for these RNA-based innovation is no residue, so that's something very attractive because we talked about it, but also the agility of it. If pests and pathogens evolve, it will take a long time to develop a new transgenic line for that mutated or evolved pest or pathogen. Here we can design very easily 10 minutes maybe takes to design, but we could have that. But also picking up the point here, as Scott mentioned and Sue mentioned as well, design of regulation and social licensing is absolutely critical if this platform has to reach somewhere.
Yes. Thank you very much, Nina. So, Michael, I'd like your opinion. We're sort of, the area we're in is the use of genotypes in their environment. So rather than selective breeding, type of DNA technologies that we can use to actually improve the performance of animals in their specific environment. And is it possible to match better the animal health and its genetic makeup for it and then the environment they experience?
Yeah, look, we've seen significant improvements in some of the sheep traits in particular. So if we think about bare breach, for example, and reducing fly strike in merinos. So that was a great example, I think, of some of the progress that can be made. And I think about some of the challenges that we are facing around biosecurity. And Dina, we might need to follow up on some of the technology you're working on, because lumpy skin disease in Indonesia, for example, is a huge risk for our industry. And it's not, foot and mouth disease is something that would be brought in by people bringing food and, other ways of, it's going to be more people related. Lumpy skin disease is around biting insects and could be carried across to Australia in the air, so it's a big risk for us. Now, I was in Darwin a couple of weeks ago, and similar conversations with APVMA deregistering chemicals that are currently used for biting insects in Northern Australia are being deregistered. So they just won't be able to be used, and we don't have enough viable alternatives. So selecting for resistant animals that can, we already have tropical breeds in Northern Australia, but there's a lot more temperate breeds heading further north. Are there naturally resistant animals to insects? And then we look at the insect themselves, and are there control mechanisms that we can put in place? But then you start looking at just the simple things like, polled animals rather than needing to dehorn. There's a whole range of aspects around animal welfare that genomics plays a considerable role in. And I think the other one is environmental sustainability. Methane's the big one, and a lot of that research is well advanced, and I expect we'll be able to commercialise methane as a trait, but equally making sure that we don't compromise some of the efficiency components as well. So we're still going to have to produce a hell of a lot of food to feed the growing population, and we need to do it in a way where we're optimising production in extremely diverse production environments, but also making sure that we can drive progress, and I think net feed intake is another really important one, and driving fertility of both our herd and flock around the efficiencies as well. So we kind of have to stack all of these components when we start looking at genomics. And the other component from a commercial perspective is commercialising a multi-breed genetic evaluations. We've done it in sheep, but there's still 3 evaluations that could potentially become 1. And on the beef side, we're not able to compare breeds at the moment because they're all off different genetic parameters, let alone the genomic prediction for some of those traits that really are going to drive commercial demand that would shift the model from a push model of designing genomic solutions to creating pull-through demand and commercial producers pushing our research agenda harder because they see the benefit in putting more selection pressure on the traits that drive profitability and also improve market spec compliance that puts more dollars in their pocket. So I think if we can get more customer focus and get the commercial pull-through, we'll see the demand for these products also grow, and then the returns for all participants in the supply chain start to be delivered. So I think that we always start at the genetic place. I think we flip that on its head and say, well, who are we doing this for? What traits are needed? And how do we provide the tools to support that? We'll see a much more connected innovation pipeline as we go forward as well.
Great, thank you. I think I'll give this question to Ben. It's from an anonymous person, Ben or Sonia, but Ben. It's regarding the question about fitness. Should we seek to understand and learn more about from natural selection? for
Yes.
our artificial selection. Yeah, so this is a good one.
Yes. I think we're getting better at fitness. We measure things like fertility. The dairy industry has a trait called longevity or survival. We're getting better and better at measuring disease resistance and so on. But there's still probably bits, we'll learn about bits of the missing puzzle that we haven't started measuring yet. So perhaps natural selection can help us there. One thing you can actually do, and I'm pretty sure the person that asked this knows this, you can look in the genome, our genomes, our livestock genomes, crop genomes, and you can actually see which regions of the genome have been selected historically by natural selection, and then when domestication came in by us. And you can see that reasonably clearly in these large data sets we've got. So maybe by interrogating that, we can learn more about when fitness was the main driver for selection, what was actually selected for.
Mm-hmm. Thank you. And I have another question that is more for Sonia. And Sonia also wanted to talk about, or I have a question for you, is to use technologies to select the right species for the right environment. And I have a question here, is like, why are we not talking about the strength of interaction between genotype and management, especially between nutrition and health? So it's this whole interplay between matching genotypes to right environments, and the question is about genotype-environment interactions in regard to nutrition and health.
Yeah. I'll probably answer this as a bit like a politician. You need to take the whole systems approach to deciding what genotype to use, and what Mick was just saying about you actually need to know your market, you need to know what the customer demand is, that you actually know what is the right genotype that produces the right product, and how can you match that with a production system that provides a sustainable and efficient production. So you need to consider all of these aspects to actually make it a lasting system that meets market expectation. In aquaculture, there's still some basic challenges, like, for example, closing the life cycle. As I said, there's still some species where wild broodstock is still used. Your genetic gain is always going to be compromised. Your biosecurity status is always going to be compromised if you still have to go out to the wild to actually get some broodstock. So first step is that actually the life cycle has to be closed.
We do have 1 minute, so...
I got 1 minute.
No, we have, as a group, 1 minute.
All right. But
think about the whole group life cycle and take it.
But then I probably want to give one really good example that recently hit the news about pompano. That is the great oyster cracker. It's a new species that CSIRO is developing as a white flesh species. And what we've actually done is, over the last few years, have closed the life cycle. We've been looking at the fish as a product, what its eating quality, which market does it suit, what does need to be improved, what production system does it actually suit. It's quite a social, quite a docile fish, so a RAS system is actually quite useful. So I think all these you have as an opportunity with a new species, but certainly also need to be considered with the existing one. How do you make it sustainable as a system?
Thank you. 2 quick questions. The first one is by Mashani. How has gene editing been used in livestock in Australia, and are there constraints from regulations? Anyone take that on?
I'll take that one.
Do that? Yep, okay.
Yeah, one of the problems with gene editing in livestock is what would you actually edit? In my presentation, I stressed that for most of our traits, there are, in fact, thousands of mutations across the genome that affect our traits like milk yield, even disease resistance, quality as well. There are many, many mutations, and none of them have a particularly large effect. So in livestock, it's actually, we've looked into this quite a lot, it's not actually that clear what you would edit. What you would edit is if you could find mutations that made animals resistant to foot and mouth disease, for example. And there's Chinese groups who claim to have discovered such mutations. But for the production traits and most of our traits, we're in a situation where it's actually difficult to work out what to edit. Things like horned and polled are more obvious examples. You could do that, but we're already making huge progress on that with our conventional selection, marker-assisted selection. So that's the current state of affairs. We have just proposed a way of doing gene editing in a massively parallel way, where you would target lots and lots of mutations simultaneously, and in plants that's happening, but it's very technically difficult to actually pull off.
Thank you. And I have one question that's sitting here for like half an hour by Gunilla, which I'll answer as the last question of the session. What are the negative effects of selective breeding, given that it cannot be reversed? So the negative effects of selective breeding is if we go too far, and we've gone too far in productivity in some aspects, and that has had negative effects on welfare and health of animals. And that's the reason why we have expanded our breeding programs to include these traits. However, it actually, if we make a mistake in selective breeding, we realize that we can reverse it. Also in regards to the productivity, the example of litter size that I gave, some breeding comes maybe in back fat, for example. There are examples where the animals have become too lean and the reverse selection is possible as well. We can breed slow-growing fat animals that basically have small litters. We can do this. We can reverse the gains that we have made in productivity, efficiency, as well as health and welfare, to go back to unimproved genotypes. And we can drive genetic trends in individual traits like a car. We can accelerate it, we can keep it constant, and we can reverse it. So this is our session. I like to thank everyone of Ben as a speaker and every panel member for their contributions, and thank you for your attention and for your detailed questions, and thank you very much.